This presentation is about when meiosis goes wrong, when errors occur and you end up with too many chromosomes or too few chromosomes, or deletions or insertions or mutations of the chromosomes. So chromosomal abnormalities can result from many different things. First of all, you can get the incorrect number of chromosomes from a process called non-disjunction. Non-disjunction is when the chromosomes don't separate properly during meiosis. So you end up with too many or too few chromosomes in each of the gametes produced. You could also have a breakage of the chromosomes. And this is when the chromosome breaks apart and then some genes are either deleted, or sometimes you can get a gene that makes too many copies of itself, and so you get a duplication. You can have a gene that switches the order that it's in, which is called an inversion. And you can have a gene that breaks off of one chromosome and attaches to a different chromosome, and that's called translocation. And all of these things end up with giving you mutated chromosomes or the incorrect number of chromosomes in the cell. So non-disjunction happens when there's a problem with the meiotic spindle, and it causes an error in the number of chromosomes in each daughter cell. So for instance, the homologous chromosomes don't separate properly during meiosis 1, or it could also happen if the sister chromatids fail to separate during meiosis 2. And you end up with either too many or too few chromosomes in a given gamete. So usually, if meiosis happens correctly, during meiosis 1, you have a separation of the homologous pairs of chromosomes, and each cell ends up with 3 complete chromosomes. Each gamete would then end up with 3 chromosomes, the correct haploid number. Now, when non-disjunction happens, the meiotic spindle might take 2 chromosomes to the same direction. This causes one cell to end up with too few chromosomes while the other one ends up with too many. So you end up with one cell that's n-1 and one cell that's n+1. It has an extra chromosome. And this can either happen during meiosis 1, in which case all of the gametes are going to end up with the wrong number of chromosomes. Some of them are going to have too many, some will have too few, but they all are wrong. If meiosis continues properly the first time, but the non-disjunction happens during meiosis 2, then you end up with some of the gametes, half of the gametes, having the incorrect number, and the other half are normal. So if the baby has the wrong chromosome number, that's either called trisomy or monosomy. Trisomy is when the cells have 3 copies of a chromosome instead of just 2. So here is an example of an egg that has too many of a certain chromosome and a sperm that's normal. When they come together, the baby ends up with an extra of that type of chromosome. If you have the instance of monosomy, then the cells only have 1 copy of a chromosome. So that's when, for instance, the egg is missing a chromosome, sperm comes in, and the baby ends up with 1 less chromosome than it needs. This actually happens fairly frequently in humans, but usually the results are so disastrous that the embryos are spontaneously aborted. So either the mother will not even know that she is pregnant, or the abortion will happen very early on in the pregnancy. Now, certain conditions are survivable, but they have a characteristic set of symptoms which makes them called a syndrome. An example of this is Down syndrome, or trisomy 21. The other 2 trisomies that can happen with autosomal chromosomes are trisomy 13 and trisomy 18, but both of those are not survivable past early childhood. Now, in the case with sex chromosomes, the X and the Y chromosomes, human development is more tolerant of having the incorrect number of sex chromosomes. There's a wide variety of distinct syndromes that can happen in humans. You can have Klinefelter syndrome, you can have trisomy X where they have too many X's. You can have Jacob's syndrome if you have an extra Y, or Turner syndrome, which is monosomy X. Have more or less sex chromosomes. That's not as much of a problem as having too many or too few of the autosomal chromosomes. So the other thing that can happen is you can have a change in chromosome structure. So if there's an error in replication of the DNA, you can end up with a gene being deleted, or you can end up with a duplication of a DNA. So there's too many copies of that one gene. You could also have an error of crossing over. So crossing over is when the two chromosomes exchange pieces of them during prophase to metaphase of meiosis 1. And you can get an inversion where the gene is put back in the wrong order. Or you can have translocation, where one piece is taken off of one chromosome but actually put back on the end of a completely different chromosome. So you move a segment from one chromosome to another.